Chapter 10. So this is our last chapter in the book that we're going to be studying, and it's on antimicrobials. Antimicrobials, you're going to dispense these drugs on a daily basis. I'm going to start this chapter talking in general about antimicrobials and a little bit about bacteria, and then we're going to start on specific drugs in this category. We'll start with penicillin, and then move on to other types of antimicrobials. I'm going to emphasize that, as I hope I have with other chapters, and that is you need to know how these different categories, how they act, but also to produce their effects, but also the things that we need to be careful of with specific drugs. So I'm going to be trying to focus on that. So, what are antimicrobials? Antimicrobials, um, this term I'm going to use interchangeably with antibiotic, and because that's very common, but specifically antimicrobials are drugs that we use to kill or inhibit the growth of microorganisms. And those microorganisms include bacteria, protozoa, viruses, and fungi. And we would refer to, other than bacteria, we would refer to these drugs as things like antiprotozoals or antivirals, antifungals. But the term antimicrobial encompasses it all. It's a non-specific term. And it also covers the kind of traditionally created antibiotics and synthesized drugs. So it's a broad term. And as I said, antibiotic equals antimicrobial. So you'll hear me use them interchangeably. The technical definition comes from how antimicrobials were discovered. And Alexander Fleming, back in the 1920s, was the one who first discovered penicillin. And he did so by being not too tidy in his lab. And so it was a happy mistake from not being very clean in his lab. He was European, and Europeans tend to take the month of August off. And he was studying Staphylococcus. He was studying a bacteria. He had a bunch of cultures in plates, and he put them into the corner of his lab and left for a month on vacation. When he came back, he discovered in some of these cultures that there were two growths, the one that he was trying to grow, and it was actually a fungus that was suppressing the growth of another one. And it's kind of illustrated by what we do today when we are looking to see whether a bacteria is susceptible to a type of antibiotic. So this is a disc. It's a plate that has an agar gel, so it's a media that will grow the bacteria. And so when we do these in a clinic, or they do them in a lab, if you send the sample out, you take a sample from what you're trying to culture. So let's say an animal has an infection, you take a sterile cotton swab, and you then gather that bacteria and smear it on this plate. And you can see that these are different antibiotic discs. So they're different antibiotics. And the yellow is the growth of the bacteria that is from the animal. And you can see that we have a very clear line of demarcation. So this antibiotic is very clearly inhibiting the growth. This one a little less so. The outlines are a little fuzzy. That bacteria is getting a little closer to it, so it's not as susceptible. The bacteria is not as susceptible to this type of antibiotic. Well, this was the same sort of thing. If you think about this being maybe the penicillin mold growing, and then this is the staph that he was trying to grow, it is being inhibited by the fungus that was producing penicillin, basically. He called it mold juice for a while, and then he decided to call it penicillin. So that was our first antibiotic. So in this case, it's natural substances that are inhibiting the growth of another um, microorganism. But nowadays, once we discover what that particular um, chemical substance is that's inhibiting the growth, we now produce it synthetically in a lab. So we don't rely on having to produce cultures of one microorganism to get the substance that will inhibit the growth of others. So antibacterials, that term just is talking about a drug that can be either synthetically or naturally derived, can have either static or cidal effects on bacteria, and we'll go over what that means in a minute. We're going to exclude in our discussion antibacterials that are used outside the body. So things like hand washes and things that you use for surgical scrubs. You hopefully covered that in surgical nursing. Um, and or your parasitology class too. Sometimes we talk about disinfectants and antiseptics in either of those classes. So the term cidal, when we say bactericidal, virucidal, fungicidal, we're talking about drugs that kill the microorganism. They are used when an animal is immunocompromised, but they're also used if they are the appropriate drug to use. So when an animal is immunocompromised and they do not have a very good working immune system, we need to make sure that we get rid of the microorganism because there is nothing there to mop them up after their growth has been suppressed. So when we use the term cidal, we're talking about an antimicrobial that will actually kill the microorganism. You will see I have these colorful pictures in this talk, and um, it's hard to, pick things to sort of illustrate my slides here. But these are actually electron microscope pictures that were then colored, and so it was sort of an art project. And they are actually pictures of the chemicals that are antibiotics. So it is related. It's just adding a little color to my slides. So on the other hand, we have static drugs, and these are drugs that do not outright kill the microorganism, but they inhibit them. They inhibit their growth, and that allows the animal's immune system to come in and get rid of them, eliminate them from wherever the infection site is. We refer to these as bacteriostatic, virustatic, fungostatic. The importance is that when we talk about these drugs, it is important to note whether they are static or cidal in their action when we have an animal that has a compromised immune system in particular. We definitely can use either in an animal that has a functional immune system. So depending on the concentration that reaches the site, we can have either effect. If the concentration is low, we can have a static effect. When it's high, we can have a cidal effect. So our goal of antimicrobial therapy is to kill or disable what we're trying to eliminate, the infection, without affecting the host. So we always have to think about our patient. Here we have a little guy, and so there's certain, just like every drug, there's certain antimicrobials we'll tend to use in these young patients or older animals that cannot take stronger drugs, and so we always have to consider that. So in particular, the successful administration of antimicrobials involves three different factors. And these are the ones that are most important to consider. We're going to consider some secondary ones too, but the most important things are that the microorganism has to be susceptible to the antimicrobial. The antimicrobial has to be able to either inhibit the growth or kill the microorganism. Otherwise, we're not going to use that. the animal has to tolerate the concentration at which that antimicrobial has to reach in order to obtain its effect. And then, kind of like a secret agent, it has to be able, the antimicrobial has to reach the site of infection at a concentration that is going to um, not harm the animal, but uh, inhibit or kill the microorganism. So those three conditions have to be met in order to for us to want to use a particular antimicrobial for a particular infection. So some other things that we're going to consider, and I'm going to talk more at length about bacterial resistance. We want to avoid that. And part of that is having good compliance. So the owner has to be able to administer the drug. And we've talked a little bit before about, in the formulary, it may say that you can administer a particular drug 2 times a day or 3 times a day or 1 time a day. And maybe the 1 time a day is going to get your client to be able to give that drug successfully and not forget about it. So we do need to think about that. We can't do it at the expense of those first three considerations of getting that antimicrobial to the site of action in the high enough concentration in particular. But it is important to remember that we have to make it not too difficult. I'm showing this picture pretty deliberately. This would be a technician, right, or a veterinarian getting this pill way down in this animal's throat. We have better ways to help owners administer drugs. This dog is going to be resistant to this if this happens to it 2 or 3 times a day, unless it's a very, very um, submissive animal. So we want to always think about how are we going to help the owner to comply with what we need to have happen with antimicrobials. So some concepts that you need to know about are the minimum inhibitory concentration, otherwise known as the MIC. The minimum inhibitory concentration is, as the name implies, the minimum concentration at which a microorganism will be inhibited. If you see this rack, it has different, these are just colors, but the concept is here of we have a very light concentration to a very dark concentration. So let's say that's our antimicrobial, and we introduce a culture of a bacteria that we want to know, will this antimicrobial be able to inhibit or kill it? And so we look for where we find the growth, and the growth is the last place where we find growth of the microorganism in the solution of the antimicrobial. The one step above where we do not see that growth, that is the one that is the concentration at which the organism is inhibited. So that's its MIC. Every bacterial strain and species has a different minimum inhibitory concentration for every antimicrobial that is out there. And so these are tested in internationally certified labs, and that minimum inhibitory concentration is determined for each antimicrobial and for specific organisms. You need to know is the MTD, which is the maximum tolerated dose. So in order for us to use an antibiotic, it needs to be below the maximum tolerated dose. So that's the dose at which a particular animal species can tolerate the antimicrobial without causing effects that are worse than the infection, let's say, or just not very tolerated. So when we have an antibiotic that has an MIC, that minimum inhibitory concentration, that's above the maximum tolerated dose, it's referred to as being resistant. We can't use it. If that minimum inhibitory concentration is below the maximum tolerated dose, then we term that as susceptible, and we can use this drug to treat a particular condition in the animal. This picture illustrates that minimum inhibitory concentration. So in this case, it would be this 1.6 micrograms per ml. You can see there's no growth in this tube, and this one has growth. So this is the minimum.